Hello everyone and welcome. In this video we're going to be checking out October-November 2021 variant 1 of basically 9700 AS Biology. This is a walkthrough of the paper. I'm going to be solving it with you guys, and of course if you have any general questions in AS or in A2 Biology, you can leave those down in the comments before we get started. Also, if you have any resources that you have found and you're studying from them and they're wonderful, please make sure to share them with other students if you'd like. This is going to help a whole lot of students that are on this channel. Let's get started. Question number 1. A student observes a cell using a light microscope. Then the student draws the cell. Which items will the student need to calculate the magnification of the drawing? In order to calculate the magnification, you have to remember the formula. The formula says, let me just change the size. Okay, the formula says it's image length divided by actual length multiplied by the magnification. And so I remember A is for actual length, M for magnification, I for image length. In order to find the image length, we're going to need a ruler because this is a drawing. In order to find the actual length, we're going to need to use the stage micrometer and of course the eyepiece critical. So eyepiece critical, stage micrometer, and ruler. No need for a hand lens, that's totally useless. So it's 1, 2, and 4. Okay. Number 2. Which equations correctly show the relationships between magnification, image size, and actual size in microscopy? So let's see. We already established that magnification is image size or image length over actual length, and so this is correct. Okay, let's look at the other formulas. This is wrong. Actual size equals to image size over magnification. Actually, actual size does equal to image length over magnification. That's again based on a triangle where I is above M, and that equals to A. Okay, actual size, however, does not equal to magnification over image size, and so the answer is 1 and 3 only, which is A. Number 3. In which cell structure does DNA transcription occur? So in chloroplasts, actually DNA transcription does occur considering there are 70S ribosomes inside. So there happens to be protein synthesis, and as part of protein synthesis we have transcription and translation. How does transcription happen? Using DNA that basically here it's circular DNA that's found inside the stroma, going to be used for transcription. Also the same thing for mitochondria and for nucleus. Okay, so it's letter D. Number 4. Which row could identify the nucleic acids present in 2 different virus particles in a sample of air? Remember that viruses can either be adenoviruses or retroviruses. What I mean by this is the virus can have either DNA or RNA inside their nuclear envelope. And so again, in the nucleic acid core could either be made of DNA or RNA, not 2 at the same time. So the first option is wrong. Okay, DNA and RNA, yes, this is known as an adenovirus, okay? And when it comes to RNA only, this is known as a retrovirus. Basically, we have to choose 2 and 3, which is letter C. 5 tests on 4 samples from a mixture of biological molecules gave the results shown in the table. Which biological molecules were in the mixture? Let's see. So boiled with excess Benedict solution, blue is a negative result, meaning that it does not have any reducing sugars. We can remove that. Okay, boiled with excess Benedict solution after acid hydrolysis and neutralization. This shows that it has non-reducing sugar because hydrolysis is going to be breaking down the covalent bonds that are basically glycosidic bonds when it comes to, but if it basically the Benedict is positive, then obviously it's a glycosidic bond that's being broken down to form the monosaccharides, which are reducing sugars and are going to be positive for Benedict's test, which is why the answer here is red. So this is going to have non-reducing sugar. An example is sucrose. Okay. Now, biuret reagent is purple, meaning it's a positive test for protein. Iodine solution is yellow, meaning it's a negative test for starch. So we should cancel out any option with starch with reducing sugar. However, we should get the answer with non-reducing sugar and protein, which is letter C. 6. 1 molecule of X is formed by a single condensation reaction releasing 1 molecule of water. What is molecule X? So 1 molecule of water should be a hint because you should know to not choose a phospholipid. It's wrong. Rather, you choose a triglyceride because triglycerides are going to release 3 molecules of water. Phospholipids will release 2 molecules of water, and that's by their names. Tri means there are 3 ester bonds, and phospholipids, basically you already know that they have a phosphate head and 2 fatty acid chains which are joined to glycerol, and that's again by an ester bond. a polysaccharide will release millions of molecules of water, not just one. So that should be a disaccharide, because it's one molecule, one monosaccharide joined together by one condensation reaction, one glycosidic bond to release one molecule of water. Number 7, the table shows the information about carbohydrate polymers, which row describes glycogen. Okay, so glycogen contains both 1,4 and 1,6 alpha glycosidic bonds, and so we should choose those. The shape is not helical, that's the shape of, amylose. So basically, not found in starch, not found in glycogen, and so basically its branched function is storage, not structure. That's the function of another polysaccharide known as cellulose, and basically chitin too, which are found in cell walls. So cellulose found in cell wall of plants, chitin found in cell walls of fungi, and so that's letter A. The diagram shows 4 phospholipid molecules. Which molecule could contribute most to fluidity of the cell surface membrane? So contributing the most to the fluidity means basically that it's going to be having the greatest effect on increasing the fluidity. And what can is basically the fatty acid with the most, fatty acid, basically double bonds. So basically the most unsaturated fatty acid. And here we can see we have 4 double bonds total, meaning it's going to have the greatest effect on the fluidity. Remember, when it is unsaturated like this, it's going to decrease the fluidity the most. This is going to affect the fluidity, but not as much as letter D. Let's move on. Which properties of phospholipids explain why single layers of phospholipids added to water immediately form bilayers? So the reason they immediately form a bilayer is because of the hydrophilic and hydrophobic properties of, the fatty acid chains and the phosphate heads. So the hydrophobic fatty acid chains repel water molecules, so the tails pack together. That is correct. The non-polar fatty acid chains are attracted to each other by hydrophobic interactions. Well, actually, yes, they are. The non-polar fatty acids are attracted to each other by hydrophobic interactions. Hydrogen bonds form between the phosphate groups and the water. 1, 2, and 3 is correct. Now, for the diagram shows 3 examples of different bonds. Which bond holds the tertiary structure of proteins together? The tertiary structure of proteins is held by ionic bonds, hydrogen bonds, disulfide bonds, and hydrophobic interactions. Bond 1 shows a hydrogen bond, because you can see a bond between hydrogen and oxygen, and it is like some weak bond which is represented by these dots. So obviously this is a hydrogen bond, so yes, it does hold it. Bond 2 shows a peptide bond, and you can see that because it's a bond between carbon and nitrogen, and it's the direct bond. And of course, a peptide bond does not hold a tertiary structure, however it does hold a primary structure. So this is wrong. Bond 3 is a disulfide bond, because we can see 2 sulfur atoms. It's supposed to be a double bond here, however it's not shown. Maybe this is a new representation of the disulfide bond. So what is it going to be? It's 1 and 3 only. Okay. 11. Which feature of collagen enables it to fulfill the structural role in skin and in tendons? Okay, so the structural role in skin and tendons is because, let's read the options. Adjacent collagen molecules are linked by ester bonds. Adjacent collagen molecules are not linked by ester bonds. They are linked, however, by covalent bonds. Collagen fibers form layers with the fibers in different directions. Well, actually, no, that's to do with cellulose. They form it in parallel directions. Okay, collagen molecules are formed as triple helices of polypeptide chains. Okay, or polypeptide chains of collagen are tightly folded into compact shapes. That's wrong. This is for storage molecules. We do not say that for something else. So that's letter C. Okay, number 12, the graph shows how the concentration of 4 components, 1, 2, 3, and 4, of an enzyme catalyzed reaction change with time. Which component is an enzyme substrate complex? Let's first look at the graphs and label them, and hence be able to determine what is the enzyme substrate complex. So for number 1, we can see starting concentration is 0, and then it increases as time goes farther. So we can assume this is the product concentration. I think it's pretty easy to analyze this because, product will be produced over time. However, what will be used up is the substrate, and we can see it's getting used up but never reaches 0, meaning that the substrate is never completely used up. So it's still in excess. Number 3 is substrate. Now, why do I say this is because this is very important in order to identify the enzyme substrate complex. Based on this, we're able to identify whether the rate is constant, the formation of enzyme substrate complex, or whether it is decreasing eventually. So let's see. We have either number 4 or number 2. Number 2 doesn't make sense considering the, substrate concentration is in excess. Why is the concentration decreasing suddenly right here? This doesn't make any sense. However, number 4 does, because we can see that the substrate is in excess, the product is still being produced at the maximum rate, and the number 4 is the rate of formation of enzyme substrate complex, which is constant and the highest it can be, like so far, So that's letter D. The table shows the Michaelis-Menten constant Km for 3 enzymes, enzyme C, P, F, and you can see the Km, as you can see, it's actually decreasing. Now, you might make a mistake here, which is basically, and hopefully not, okay, it's basically that you think that is a positive power and hence the number is larger. However, you can see it's negative, and that means it's actually a much smaller number. So for F, it has the lowest Km value, and that's why the answer should be C. Enzyme F has the greatest affinity for its substrate. As you decrease the Km value, you increase the affinity of the enzyme for its substrate. Now let me explain this for you. Say you have the graph of the substrate concentration against the rate of reaction, which is a very familiar graph, and here I added another line, which is basically showing as you can see, the substrate concentration against the rate of reaction, but when a competitive inhibitor is added. And when a competitive inhibitor is added, you can see that actually the Km has increased. So this is, we know what the Km is. It's the substrate concentration which have the available enzyme active sites are fully occupied. Right, and so basically if we look at this, here we have, this is the Vmax, okay? At half the Vmax, we have the Km. Now, in order to look at, this is the same Vmax, half the Vmax is going to be right here. You have a greater Km value. A greater Km value means, as I mentioned, a lower affinity. And this is proven here because when you add a competitive inhibitor, you do not have a strong attraction between the enzyme and substrate. At least not as strong an attraction if you left the substrate and enzyme alone. Because obviously, if you leave two things alone, they're going to be, more compelled to talk to each other. They'll be more bored. However, when you have a third party member, they're going to be, occupied with its presence, right? And so it's not going to be such a strong attraction. And this is what affinity means. It's the attraction of the enzyme for its substrate. And so you can see that here the attraction decreases because there is a competitive inhibitor. And what happens when the attraction decreases? The Km increases. So same thing should be correct in the opposite direction. And when you decrease the Km, the attraction must increase. The affinity, as I mentioned, must increase. And that's why the answer is C. Okay. What explains how a signal molecule produced by one cell can be detected by a target cell? So the reason it can be detected is because the signal molecule has a complementary shaped receptor to it either on the surface of the cell surface membrane, or within the nucleus or the cytoplasm, depending on the type of signal molecule. If the signal molecule is, maybe a steroid, for example, or something that is basically not a protein, not hydrophilic, okay, this is going to be able to cross the cell surface membrane like estrogen or something and move into the cell and bind to the complementary receptor, whether in the nucleus or the cytoplasm, okay? However, if it was a protein and it's hydrophilic and it's large and it cannot cross the phospholipid bilayer and the hydrophobic, inner core of the hydrophobic fatty acid inner core of the phospholipid bilayer, it's going to have the complementary receptor on the surface, and when it binds, it's going to change the shape of this receptor, leading to an enzyme cascade. Now let's go ahead and look at the options. A signal molecule can bind to any type of cell surface receptor. That is wrong, okay? He mentioned why a single signal molecule can be basically produced by one cell, can be detected by the specific target cell. This does not make sense. A signal molecule has complementary shape to a cell surface receptor, correct? Signal molecules enter the cell by diffusion, not necessarily, not always, and this is not the reason they are detected by target cells. Same thing for endocytosis, so that's why the answer is B. 15. Some enzymes are produced in cells of the pancreas. The enzymes are secreted when required. Which process is used to transport these enzymes out of the cells of the pancreas? Enzymes are going to be secreted when required, and so that's going to be released by exocytosis. Enzymes can't just, move across the cell because they're going to be digesting stuff, maybe, and they're mostly going to be produced inside the Golgi apparatus. Always going to be modified inside the Golgi apparatus, and the Golgi apparatus is going to be releasing its contents inside the secretory vesicles or the Golgi vesicles. And these Golgi vesicles are going to move across the cytoplasm by the microtubules of the cytoskeleton towards the cell surface membrane, where it's going to bind and release its components by exocytosis, which is an energy consuming process. Okay, and I'm gonna be doing a drawing here for you. This is a Golgi apparatus, let's say. Okay, it's going to be releasing these Golgi vesicles that contain the enzyme, which is going to move across the cytoplasm again, guarded by the microtubules of the cytoskeleton, and fusing with the cell surface membrane, releasing its contents, and I hope you can imagine that. Okay. Plant cells were submerged in a solution with a water potential less negative than found inside the cells. So here you immediately have to draw what they mentioned. So again, it is submerged in a solution that is less negative than inside the cell. So inside the cell is going to be more concentrated than outside the cell. Okay, so here we can see that it has a lower water potential, however outside it's going to be a higher water potential, and this is in the plant cell. Okay, and basically what describes the condition of the plant cell after 20 minutes. So we can understand that water is going to move from a region of higher water potential to lower water potential down a water potential gradient through a partially permeable membrane by osmosis, meaning it's going to be moving inside. Now when it moves inside, obviously this makes the plant cell turgid. Initially it wasn't so turgid, it was like maybe that same sort of flaccid. Now it is turgid. It doesn't burst because it has a cell wall. It's not incipient plasmolysis because, well, again, it's gaining water, not losing it. Same thing with plasmolyzed. Okay, 17. What will be present in each chromosome at the end of G2 phase of the cell cycle? So G2 happens after S phase. And S phase is basically the phase where there is, synthesis, or let's say replication of the chromosomes. Okay, the chromosomes will be duplicated, and we say it's not really duplicate, more like, chromosomes are going to have 2 chromatids instead of 1. And so 1 centromere and 2 chromatids. Okay, let's see. So basically two centromeres. No, this is wrong. 2 centrioles. There's no centrioles inside chromosomes. 2 molecules of DNA. This is correct because 1 molecule of DNA is equivalent of 1 chromatid. Okay, and 2 telomeres. No, there are 4 because the telomere is the end of the chromosome, and you can see here we have 4 ends. Okay, and that is letter C. Okay, what events listed are part of the cell cycle? We have interphase, metaphase, cytokinesis. So part of the cell cycle, not part of mitosis. Mitosis will include, metaphase. Okay, this is basically including PMAT, meaning prophase, metaphase, anaphase, and telophase. Okay, cytokinesis and interphase are overall part of the cell cycle, and so it's 1, 2, and 3. However, if they asked what is part of the mitotic cell cycle, or let's say what is part of mitosis, you'll only choose number 2. Okay. Which features of an organism are affected by a drug that kills cells that are divided mitotically? Interesting. So we have cell repair, cell replacement, number of stem cells, tissue repair, tumor formation. Okay, so let's see. The functions of mitosis are actually not cell repair. Okay, it's cell replacement. Okay, because mitosis is going to be cell division. Cell is going to divide and basically replace the damaged cell. However, it has nothing to do with what's within the cell. Okay, number of stem cells will be affected because stem cells are produced by mitosis. Tissue repair is going to be affected because it requires cell replacement. And when there is no cell replacement, then the tissue will not be repaired. Okay, so this is correct. Tumor formation, same thing, because the tumor is going to be formed by the continuous cell division of the, cancerous cells by mitosis. And so it's number 2, 3, 4, and 5, which is letter D. Okay, so for number 20, the graph shows how the distance between sister chromatids, curve P, and the distance between chromatids and centrioles, curve Q, changes during part of mitosis. And, okay, so we have part of mitosis. This is very important. So which statements could be correct? T to V represents interphase. This is wrong because interphase is not part of mitosis. T to W represents metaphase. Yes. W to X represents anaphase. Correct. So it's 2 and 3 only. However, another things that basically maybe you might make this mistake. Maybe you didn't focus that there is mitosis written here. However, you have to understand that centrioles are going to only be available at the opposite poles of the spindle when prophase is done, because this is basically the point at which they migrate towards the opposite poles of the spindle. Am I right? Or towards the opposite poles of the nucleus because they didn't form the spindle yet. And so basically, you cannot say that T to V is interphase because we still do not have, the centrioles at a specific place where we can correctly compare the distance, considering we only have 1 centriole, number 1, and we do not have them at the opposite directions. And so that is basically why the answer here should be B, and you should cancel out number 1. It helps to explain why the 2 sugar phosphate chains in DNA molecules are constant distance apart. So why the 2 sugar phosphate chains aren't always a constant distance apart is because, the bonds between, the basically adenine, thymine, guanine, cytosine is always going to be the same size, considering that it's a bond between a purine and a pyrimidine. A pyrimidine is a single ring structure, a purine is a double ring structure. And purines could be either, adenine or guanine. However, pyrimidines are either cytosine or thymine. And because cytosine is going to bind to guanine and adenine binds to thymine, that means basically the distance is always going to be the same. Okay, so adenine and thymine are held by the same number of hydrogen bonds, cytosine and guanine. Okay, each nucleotide molecule is the same size. That's wrong. We have purines and we have pyrimidines. Each purine base is linked to a pyrimidine. That is the exact reason. Okay. During DNA replication, what must happen before a newly added nucleotide is bonded to the next nucleotide in the strand? Let's see. Complementary base pairing, hydrogen bond formation, and phosphodiester bond formation. Now, phosphodiester bond formation Okay, no, this is going to be happening with like when basically the newly added is going to be binding. So this is not before. Okay, hydrogen bond formation, yes, and complementary base pairing, yes. So it's 1 and 2 only, which is B. Okay, well, an antibiotic enters bacterial cells through a membrane channel protein P, and some bacterial cells have shown resistance to the antibiotic by acquiring a mutation which alters P. This mutation prevents the entry of antibiotic to the cell. Which conclusions can be drawn about how resistance to this antibiotic developed in these bacteria? Okay, so the resistance basically is going to be happening by a mutation, as they mentioned. The mutation changed the order of amino acids in the gene coding for P. This could be possible, yes. Okay, basically the mutation resulted in the production of P with an altered tertiary structure, and because maybe it changed the primary structure, changes the altered tertiary structure, that is correct. The antibiotic is a hydrophobic molecule and so cannot cross the phospholipid bilayer to enter the cell. That is wrong. It does not have anything to do with, a mutation. The mutation does not alter the antibiotic, it alters the genes of the bacteria itself. Okay, it does not happen outside the bacteria, it doesn't happen to the antibiotic. Okay, and if it was initially hydrophobic, it would not have been able to, cross the channel, because the channel protein is like, pathway for hydrophilic molecules, number one. Number two, if it's hydrophobic, then it can cross the phospholipid bilayer easily and without any need for a channel protein. 1 and 2 only, which is basically not an option 2, so let's choose 2 only, because the mutation changed the order of amino acids in the gene coding for P. The gene does not change, the gene does not have amino acids, okay? And the gene has only nucleotides, so it changes basically the order of nucleotides, let's assume, and that results in changing of amino acids. Okay, but I would assume basically that the mutation results in the production of P with an altered tertiary structure, and that's the answer. Okay, so what is correct tRNA anticodon coding for amino acids proline? So proline, here we can see the DNA triplet code is GGT, so it's going to be GGU. Because remember, when it comes to tRNA is only going to be the same thing as the DNA code, except with of course, thymine that is not found in RNA. We only have uracil, and so we have to change thymine for uracil and choose, it's the same thing, basically GGT, GGU, except for T and U. Let's move on. The table shows observations made by students about 3 different cell types P, Q, and R, as seen in transverse sections of plant stem when using a light microscope. So the ratio of the cell wall width to whole cell width, and we can see the structure observed in the cell contents. And we can see this is 1 to 10, 1 to 20 regular pattern of circles in some cells, 1 to 20 granular appearance of some cells. And this is across plant stem. So we have the xylem, the phloem, and the companion cell. So let's see which one is which. P has nothing, so I'd assume it's a xylem vessel element because it's a hollow structure, right? Q, let's see, we have other companion cell of phloem sieve tube element, and considering we can see a regular pattern of circles, I would assume that's the phloem sieve tube element, because the granular appearance should be of a cell that has, like a lot of cytoplasm, a lot of organelles, like the companion cell. Okay, and that's letter D. Water molecules are attracted to each other. This property is important for the upward movement of water in the xylem, which term is used to describe the attraction of water molecules to each other, cohesion. The diagram shows a model to demonstrate mass flow. In a plant, what are the structures W, X, Y, and Z, and what's the direction of flow of a solution along W? Okay, so the phloem is, so let's see. First off, he said that there's a concentrated sucrose solution here and a partially permeable membrane, and here we have a dilute sucrose solution. So supposedly we're going to have, sucrose moving in this direction because it's going to move from region of higher concentration to lower concentration. That's within the phloem, okay? And so basically the phloem is going to be, W. Okay, the phloem will be W. The xylem is going to be X because it's moving, the water is going to be moving across the xylem, okay? And then roots are going to be Y. Well, let's see. Concentrated sucrose solution actually is going to be at the leaves, not at the roots, because the roots are going to be, just absorbing water and stuff and not making sucrose. Sucrose is going to be made, from glucose, which is formed by photosynthesis at the leaves. And so Y is the leaves, roots is Z, and here we have W and X, and direction is from Y to Z, as I mentioned, as letter B. The parts of the heart that control heart action are listed. Which row is correct for atrial contraction and ventricular contraction? For atrial contraction, let's see. Sinoatrial node, atrioventricular node, or pericardial tissue. We have atrioventricular node produces wave of excitation. That is wrong. This is required for ventricular contraction. Pericardial tissue, again, this is required for ventricular contraction. Sinoatrial node and AV node are going to produce waves of excitation. This is not important for atrial contraction. Sinoatrial node produces waves of excitation. Correct. Okay, ventricular contraction, pericardial tissue carries waves of excitation. Perfect. Letter D. The diagram shows parts of the circulatory system in a mammal. Where is the blood pressure and the speed of flow the lowest? Okay, the blood pressure, let's see, the lowest blood pressure is going to be found inside the capillaries because, well, no, actually, I'm sorry. The lowest blood pressure is going to be found inside the vena cava. Here we're going to have the highest blood pressure, and then it's going to decrease, as the, like the aorta like size is going to decrease because like less blood is going to be carried. However, it still is going to be higher here than in this region because once like they're moving here in such a tiny space, it's gonna be having a high pressure, and then all of a sudden it increases, improving the flow, decreasing the blood pressure. And so here we have the lowest blood pressure inside number 4. Okay, and the lowest speed of flow is going to be inside the capillaries is number 1 because it's only 1 red blood cell passing per second. Okay, the photomicrograph shows human blood cells. What type of blood cell is X? So let's look at the cells available, and like letter X is going to be a phagocyte. Okay, so we have either a monocyte or a phagocyte or T lymphocyte or B lymphocyte. Okay, first off, we can cancel out the B lymphocyte because B lymphocyte is supposed to have the star nucleus, which is this one. A monocyte is supposed to be very large and having a kidney-shaped nucleus like this one, which is okay. And phagocyte is going to have a lobe nucleus. That is totally right. A T lymphocyte again is it's gonna also be a T lymphocyte, so it's gonna be looking something like this. So it's letter C. Okay. The diagram shows some of the reactions of carbon dioxide when it enters the blood from cells in a metabolically active tissue. Which reactions is catalyzed by the enzyme carbonic anhydrase? So remember, it's going to enter the cell. The reaction does not happen outside, it happens inside. Okay, and the reaction catalyzed here is letter C. It's carbon dioxide and water forming carbonic acid. The graph shows the oxygen dissociation curve for hemoglobin in animals that live at higher altitude and animals that live at lower altitude. What explains the oxygen dissociation curve at higher altitude? So hemoglobin has a higher affinity for oxygen. That is correct because at higher altitude there's not much oxygen available. Neither is there carbon dioxide, but obviously because there's not much oxygen available, the hemoglobin has to catch hardly onto whatever oxygen it can find, and that's by having a higher affinity. Okay, hemoglobin releases oxygen more readily. No, obviously not. Okay, the change in partial pressure of carbon dioxide causes a Bohr effect. No, the decrease in percentage of carbon dioxide causes the curve to shift to the left. No, because there's a decrease in both percentage of carbon dioxide and oxygen. So it's not just that carbon dioxide is the major factor here, it's not. Okay. 33. The diagram shows 3 features found in mammalian gas exchange system. Which structures of gas exchange system could be represented by the X in the diagram? Okay, so serrated epithelium, goblet cells, cartilage are all found inside the trachea and bronchus. Okay. What are short-term effects of nicotine on the cardiovascular system? This is not part of the syllabus anymore. Okay. So, some features of a disease are listed. It can be transmitted by animals to other animals, including humans. One mode of transmission is by transfusion with contaminated blood. The causative organism can show multiple drug resistance. The majority of humans who die from the disease are children. Which disease has all these features? So let's look at this. Cholera is not correct because the mode of transfusion, this is obviously wrong. Okay, HIV or AIDS, well, really, when it shows multiple drug resistance, we don't really have much drugs against HIV. We couldn't really produce anything much against it, and so we can't really assume that. However, we can assume malaria because malaria does have multiple drug resistance, and it also can be transmitted by blood, by basically transfusion via mosquitoes, and also by transfusion when it comes to actually transfusion with blood. Okay, and it can be transmitted by animals to other animals. Yes, it can be transmitted from mosquitoes to dogs. Tuberculosis, well, no, that's wrong because, well, we can't assume that is by blood. It has nothing to do with blood. And also, basically, the majority of humans who die from the disease are children. Yes, this obviously basically goes around with malaria too. The following advice was given to a person traveling the world or traveling to a country where there had been an outbreak of an infectious disease. He says, cook food well and eat it hot, peel fruit and vegetables, drink only cool boiled water, wash hands often with soap and cool boiled water. What infectious disease would this advice have to protect against? So I would assume that you are going to be protecting yourself against cholera. That's letter A because it only has to do with food, and that's it. Okay, which description of multiple drug resistance in bacteria is correct? Bacteria have the DNA which are resistant. Which description? Which description of multiple drug resistance in bacteria is correct? Bacteria have DNA with resistant genes for several different types of antibiotics, or different species of bacteria have DNA with resistant genes for one type of antibiotic, or large numbers of one species of bacteria are immune to several different types of antibiotics. Okay, so basically, multiple drug resistance in bacteria is basically when we have lots of like basically use lots of antibiotics, and then bacteria become resistant against all these different types of antibiotics, right? And so bacteria have DNA resistant genes to several different types of antibiotics, is letter A. Okay, 38. Which description of a T lymphocyte is correct? They're only found in blood and secrete cytokines in response to infection. Well, they can leave the blood and accumulate sites of inflammation. That is correct for T lymphocytes. They can leave the blood and secrete cytotoxins when exposed to bacteria. They circulate the blood and always present antigens. So they're actually not only found in the blood, they're found in tissue fluid as well. Okay, and basically this is a correct statement. They can leave the blood and secrete cytotoxins when exposed to bacteria. Well, yes, but that's basically like not the main thing that they do. Okay, they circulate the blood and always present antigens to response to infection. That is wrong. This is the function of antigen-presenting cells or macrophages when they are encountered with, or let's say monocytes when they encounter a pathogen. Okay, what are antibodies found? Where are antibodies found during an immune response? On the surface of memory cells. Okay. Yes, on the surface of memory cells, in blood plasma as well. Yes. Okay, that's letter A. Monoclonal antibodies are used in diagnosis and treatment of disease. They're produced using a technique known as cell fusion, which two structures are fused together during this technique. We have a cancer cell, and basically it's fused with a plasma cell. Okay, basically, letter C. And this is it for this variant. I hope this video was useful. Again, if you found it useful, please make sure to share it with the other friends, which make my find also useful. And also, as I mentioned, leave any questions that you have, any resources that you'd like to share, all down in the comments. Thank you for watching, and goodbye.